Okay, good evening, everyone, and good morning to those of you that are joining us from Japan as well. So excited. First, welcome you to Chicago and to this Society for Neuroscience for 2024. This program is one of our flagship programs. The U.S.-Japan Cooperative Program promotes scientific exchange, scientists exchange, as well as training and research collaborations between neuroscientists from the U.S. and Japan. The U.S. component of the program awards administrative supplements to currently active research grants that are supported by one of the participating NIH institutes or centers. The BRCP is a bilateral partnership with the National Institute of Physiological Sciences in Japan. Physiological Sciences in Japan. In a moment, I'm going to introduce my colleague from Japan who's going to speak. But I list here, we just recently, in 2020, from 2020 to 2022, we went through a strategic planning process for health equity at our institute, at the National Institute of Neurological Disorders and Stroke. We had a series of 10 manuscripts that were published in neurology outlining our work with recommendations from our working group of our National Advisory Council. We're actually currently going through the same process for global health. And so we had a request for information that was published, and hopefully you all were able to submit information on how we can work together to support the mission of decreasing the burden of neurological diseases globally. We're going to have a workshop actually in October of 2025, and we hope it'll be hybrid. We're going to have some people in person in Washington, D.C., but also there will be an online component. So hopefully we'll be able to have some of you to participate and give us information at that time. So you can get more information from our office from this QR code that we have listed on the slide. But today we're here to talk about the U.S.-Japan program. And I'm going to introduce my colleague from Japan, Dr. Masaki Osada. Dr. Osada is the new co-chair of the BRCP Joint Committee and professor in the Division of Behavioral Development at the National Institute of Physiological Sciences in Akasaki, Japan. Dr. Osada completed his postdoctoral fellowship at the NIH at the National Eye Institute. So now I'm going to turn it over to Dr. Osada.
So my name is Masaki Isoda, and I'm a professor of the Department of Systems Neuroscience at the National Institute for Physiological Sciences, NIPS. And I'm co-chair of the BRCP Joint Committee from this year as a successor to Professor Sadato, who played a significant role in operating the BRCP for more than 20 years. Let me briefly introduce the outline or the history and outline of Japan component of the BRCP. The BRCP was launched in 2000 with an MOU between NINDS-NIH and the NIPS under an umbrella agreement of the cooperation in research and development in science and technology. And this photograph was taken at the signing ceremony in 2000. And the person on the left in the front row is Dr. Gerald Fishback, the director of the NINDS at that time. And the person on the right is Dr. Kazuo Sasaki, the director of the NIPS at that time. And I think the person in the middle is Dr. Mark Hallett, and the person behind Dr. Fishback is Dr. Robert Bachman. I know Robert Bachman because he was a provost at the Okinawa Institute of Science and Technology when I joined there as a PI. So many people are connected. As Dr. Benson explained, the BRCP promotes scientist exchange, training, and research collaborations. And the BRCP supports all areas of brain sciences, ranging from molecular and cellular to the systems levels, both in physiology and disease. And the Japan component of the BRCP has 4 categories. One is researchers dispatched to laboratories in the United States to support younger researchers. And the second one is group joint study conducted by prominent neuroscientists. And the third one is information exchange seminars, which is actually holding workshops. And the last one is training courses dispatched to the US. And the last one is intended exclusively to support PhD students. And I believe that the BRCP has been and will be a valuable platform for the promotion of real collaborations and partnership between neuroscientists from Japan and US. And thank you very much for your attention. Maybe someone needs to switch.
Thank you. Stop sharing. So thank you, Dr. Asada. Yes, this partnership is over 20 years in the making. It's I think we're probably at 22 years that we've been working together and having this scientist and scientific and research exchange. So we're really excited. Now we're going to go to our first speaker. Our first speaker is with the All of Us research program at the NIH. And the All of Us program, Mr. Chris Lunt will talk more about the program. And we thought it was something that was really exciting, and they wanted to do outreach to the people of Japan. So Mr. Chris Lunt is the Chief Technology Officer for the All of Us research program, where he leads the building of platforms that engage participants and enables researchers. All of Us is enrolling at least 1 million diverse participants to support a research platform that accelerates precision medicine. His experience spans work in the government and commercial sectors, including founding venture backup startups. He is a lifelong learner who hosts a science book club as well. So now I'm going to turn it over to Mr. Chris Lunt.
I am Chris Lunt, the Chief Technology Officer for the All of Us Research Program. All of Us Research Program no Saiko Gijutsu Sekinincha desu. Ranto to moshimasu. Dozo yoroshiku onegaishimasu. And that's all the Japanese I'll use for this presentation. But I did live in Japan 30 years ago for a year as a student, and always welcome the chance to find ways to work again with Japan. And I'm here today to talk about the All of Us Research Program, which has put together a very large set of data that I think is going to be very useful for research going forward. And we're trying to make this as broadly available as possible. The mission of the All of Us Research Program is specifically to accelerate health research and medical breakthroughs for precision medicine through 3 main efforts. One is to go out and build a partnership with 1 million participants that will last for a long time, and that should be a very diverse group of people that fully reflects the diversity of the United States. Second, it should then collect one of the largest and richest biomedical data sets that is available and secure. And then finally, that we would make that data available to an ecosystem of researchers and communities who can make that a part of research going forward. And I'm here today to talk to you about where we are in that process. We first started in 2016, and now we have over 800,000 participants who have registered with the program. And of those, more than 500,000 have done all the basic steps of the program, which means that they've given us biosamples, which include blood, urine, and in some cases saliva, that they have filled out basic surveys that give information about their demographics, about their basic health, about their habits, and about the family history of health. And we have collected electronic health records for more than 400,000. So these are clinical records about the participants, and that we have biosamples at this point for nearly 600,000 people. And that, as I'd said earlier, this is really meant to be watashi tachi zenin desu. It's supposed to be all of us. And so the goal is to have at least half of the participants be people who are not white. And at least 80% of the participants be people who are underrepresented for some reason, whether that is their age, their income, they may be a sexual or gender minority, they may be from a geography that's very remote and hasn't participated in research in the past, to try and get a much better sense of the genomic and cultural diversity of the country. We do this by partnering with many different groups. We have a large consortium of health provider organizations, of health technology organizations, including genome sequencing centers and biobank, and also by engagement with community partners, people that can reach out to these communities that have not been part of research in the past. So we work with many groups like the Delta Research and Educational Foundation, which help us reach out to African American communities, to the Asian Health Coalition, PrideNet, which helps us engage with sexual and gender minorities, and many more. At this point, we are on version 7 of our curated data repository. So this is the data that is available for research right now. And this version has surveys from more than 400,000 people. It has 245,000 whole genome sequences, making us the largest broadly available whole genome sequence dataset other than the UK Biobank. We have more than 300,000 genotyping arrays. And in a really interesting set of work we've been doing, we've released more than 1,000 long read sequences. So most whole genome sequences are done as short read sequences, which means it's very hard to tell structural variation within the genome. So this could be inserts, deletes, copy number variation, or other things that are hard to tell in short read situations. In addition, we do collect some physical measurements at the time that people enroll in the program, including hip and waist circumference, height, weight, blood pressure, and other measures. And we have electronic health records for more than 280,000 people. And we also have Fitbit records for more than 16,000 people. And in version 7 of the repository, for the first time, we released sleep data. This is data that most researchers have never seen before and is giving us some interesting insights into people's health. And in general, the overlap between all of these data sets creates opportunities to find new discoveries about the relationship of these different measures in terms of understanding people's experienced health. And in the version that's going to be coming out in 2025, we are for the first time including some data that are cognitive measures. So you can see here, this is a guess the emotion activity. And so we go through and show a series of faces of many different races of people and have people estimate what is the emotion that the person is showing through their face. This is one of 4 activities that we have put into our program, working in collaboration with the National Institutes of Mental Health. And so the 4 cognitive tasks we have right now, one is people are shown photographs at increasing speed, and they have to say whether it's a city or a mountain. There's guess the emotion. There is a risk tolerance activity called now or later, and there's a left or right arrow activity, which is in part about people's ability to perceive again at speed. And these are all tasks designed by NIMH to try and get more base level cognitive measures. And since we've launched this, we've had more than 60,000 of each of these different kinds of tests data gathered from nearly 30,000 unique participants, which far exceeds what we had collected with NIMH before this date. And so I think it's particularly interesting to think about the correlations that we may be able to find between people's basic cognitive measures and their genomics. And I mentioned earlier that we have more than 300,000 arrays. That's the largest set of people that we do an array for everybody. And then we have 245,000 short read whole genome sequences, which includes some basic inserts and deletes. Then we have short read whole genome sequences where we include broader structural variations, including insertions and deletions, interspersed duplications, translocations, and others, and more than 1,000 people where we've done long read sequences. And that long read sequencing was done exclusively on an African-American audience, a group of people that has been very understudied in the past. The next version of the data set, which will be coming out this year, will include more than 2,000 people with short read sequences. The way that access to this resource works is that we have different tiers of access that are available. There's a public tier of summary statistics, and that's available for anybody to go look at right now. You could go, for example, and say, how many people do you have that have a diagnosis in the past of hyperemesis gravidarum? And then you would see the number that were in the data set with some basic breakdowns on things like gender and age as well. And then we have a registered tier. And to get access to the registered tier, you need to be part of an institution that signs a data use agreement with us. And then you are able, and you have to go in as an individual researcher, have identity proofing done so that you can demonstrate that you are who you say you are, and take a short 45-minute responsible conduct of research training module, and then sign a code of conduct saying that the work that you will do will not be stigmatizing or other negative sorts of research. And then you are given free access. You're not required to have an IRB. This is not human subjects research designated data, which means that you can just go in and explore the data without having a specific hypothesis. In that registered tier, you have access to the surveys, the clinical data, the electronic health record data, the physical measures. And then we also have a controlled tier. To have access to the controlled tier, it's the same things you do in the registered tier, but there's some additional parts of the responsible conduct of research training that you have to do, because at that tier you get access to genomic data. We are also planning in the future to provide access to individual biospecimen and participant data. We have started some studies now working with other groups where we are pulling biospecimens and sending them to them for them to run their own assays on that. So where there's interest, you can come and talk to us, and we're working on accelerating the number of those kinds of studies that we can do. The researcher workbench itself is where you do work on this data. It's a cloud-based resource because moving around 300,000 or 245,000 whole genome sequences is something most people don't have the capacity to do. It is a system that runs largely as Jupyter notebooks with R and Python. We do have SAS available now on the platform, as well as RStudio. And what's interesting too is when we started the program, we were thinking we were going to be collecting data and we would have data from that point going forward. But in the course of collecting the clinical data, we were able to get clinical data from many participants that went back further in time, working with the hospitals to pull that data. So we have data of some density going back all the way to 2000, and then of lighter density all the way back to the 1980s. So there's already some capacity to do pediatric research. At this point, we have just started enrolling children as well. So going forward, where there will be more children in the study, but there's already some ability to do that now. Also interesting is that we also have Fitbit data that predates the program as well. We have Fitbit data that goes back before 2015. We first launched the availability of the research platform in 2020 in a very basic form. We've been adding to that, adding genomics in 2022. And as you can see from the graph on the right, we have seen an acceleration in the number of research papers that are coming out of our data. We've had more than 300 papers come out this year alone. It has been accelerating. It's harder for me to keep up. I'm trying to read and know what they all are about. But it's exciting too to see that we've moved beyond the early papers, which were just descriptive, into active discovery. I don't remember if I included a slide on it. No, I don't think I did, but I'll mention quickly one of my favorite papers so far there is a gene that is associated with kidney disease in African Americans. This was already known, but we found is that if in a case where the person had only one copy of the gene, if there's a specific variation they could have for the other copy of the gene that would cancel out the bad version of the gene. So something that looks like a druggable target and something that's actively being explored right now. If you look then at the range of diseases that can be studied with the data set, I wanted to particularly highlight here for this audience the conditions that are in the mental health domain. So in that second column on the right, depressive disorder, bipolar disorder, dementia. We have a large number of participants that have these specific diagnoses from their clinical records. And so there's an opportunity to then explore with the other data that we have in there how things like genomics and others may contribute to those conditions. I have the slide here just mostly to have the QR code, and I'll make the slides available afterward to show again what it would take for you to be able to get onto the platform. It is pretty easy. We found that once the individual institution signs the agreement, it is a matter of weeks or days before people are already on the platform and able to make good use of the data. The data is available internationally. It is also available for commercial organizations. And everybody who comes in right now gets $300 of compute credits that they can use at the time that they start. Depending on our budget next year from Congress, that may not be true. So now's a good time to get in and see if you can secure some compute credits before those become more scarce. In looking at how this data can be used, it's been interesting to see the ways that different communities of researchers have already started to use this. They've used it in terms of developing AI ML models. They have used it for risk stratification and predictive analytics. They've used it for phenotype algorithms, for novel method development, for doing basic descriptive work of the larger populations. They've looked for associations between disease. There have been many papers already about health disparities. And with a lot of interest in particular in the historically underrepresented populations that are a part of our program. Our program also returns some genomic information back to participants. So anyone who signs up for the program receives back pharmacogenomic information, which is right now, I think, 7 different genes that are related to drug metabolism that they're told about, as well as being given some ancestry and traits information. And if they have one of the ACMG 59 variants of their monogenetic diseases, so diseases where if you have a variant in a single gene that you will develop a disease, we reach out to and offer genetic counseling to our participants to help them to try and address that issue. We are working on a number of new data types that we're bringing in now. We are running a program in conjunction with NIDDK, which is the kidney part of the NIH, on precision health. And so we are currently enrolling a set of participants that are going to have a food diary to record what it is that they're eating. And we will be collecting additional information from them, including biosamples. We have some people who will be put onto a controlled diet. So they will be sent food, they will eat only that food, and we will also biosample them. And then an even smaller group that will be in a domiciled study, which means that they will go live in the hospital for a short period of time so we can measure everything about their metabolism. Out of this program, we will be producing our first set of metabolomic information. And in addition to this, we are currently working on putting together a proteomic and transcriptomic data set as well that we anticipate will be available in 2024 or in 2025 or 2026. And that's it. And I think I brought myself in under time. I don't know if we are able to take questions here, but if we are, I will open up and thank you everybody for giving me this time. I hope this is a resource that you can use in your work. And I'm excited about the possibility of future collaborative studies.
So thank you. Thank you for that presentation. We do have about a minute for questions, if there are any questions before we move on. Does anyone have any questions for Mr. Lunt? Okay, I don't know if you're going to be able to stay on a little longer if people think about it, Chris, but
Yeah. I'll be in the background for a little bit.
Okay, very good. So if you think of something, we can go on. So now I'm going to hand it over to Dr. Sada to introduce the next speaker.
So now it's my great pleasure to introduce a prominent investigator from Japan, Dr. Kazuhiko Seki. Dr. Kazuhiko Seki. Dr. Seki is the director of the Department of Neurophysiology at the National Center for Neurology and Psychiatry in Tokyo. Dr. Seki obtained his PhD degree from the University of Tsukuba in Japan and worked as a postdoctoral research fellow at the University of Washington in Seattle. Okay. He then worked as an assistant professor at the NIPS before having this the current position. And Dr. Seki's primary research focus has been on the neural basis of bodily movement. And his expertise is basically electrophysiology, but is also actively incorporating advanced technologies such as optogenetics, viral vectors, and machine learning. And with the support of the BRCP, Dr. Seki carried out a group joint study with Dr. Ayman Azim at Salk Institute from 2019 to 2022 in order to investigate the neuronal mechanisms of sensory gain modulation during movement. Today, he will be talking about the outcomes of his successful collaboration. Dr. Seki, please.
Thank you very much. My name is Kazuhiko Seki, coming from the NCNP, Japan. Thank you very much for kind introduction for myself. Today I'm going to talk about sensory gain modulation during movement, cortical and subcortical neural mechanism. So before that, organizer asked me to include some topics before or after this talk. So I included. First one is the value gained from the BRCP partnership. It takes some time because I think this is the third time I got support from BRCP. First one is just after getting a PhD, just after finishing a postdoc at the University of Washington, his efforts, and I got a position in the Seiriken and IPC, NIPS, but still there's some work which cannot be done at Japan has been left in the University of Washington. So I applied the grant and this program kindly support me for the finishing my study at University of Washington for half a year, I remember. So that is the first time. And second time, I applied a collaborative program with, again, Dr. FS. And this is taken during that period. So he kindly visited us, and then we discussed and also writing a paper together. We wrote one paper during his stay. And also we did some, discussion about the experiment. So this is how time. But this is grant is rather earlier in my career. But starting with this career, we keep collaborating. Like this 10 years later, and now I invite him to one of his career or in my institute at the symposium. We gathered many younger scientists. And then finally, this year, he retired. So we keep communicating and keep discussing about this, our project. That is thanks for this program. And actually, as a result, this program support all of my career. I have to say thanks very much for you, organizer, or planning this grant. And then third one is this grant is actually I collaborating with Ayman Azim, who is a young PI in Salk Institute. But when we start this program, it's under the COVID, just after starting a COVID. So even if in Japan side, this grant supports a travel to US to interact each other, but it's impossible to go to US. That's difficult. But we keep discussing by using a Zoom, and thanks to program coordinator that they kindly extend the period to use this grant 1 year. At that time, I finally visit to the Salk Institute, and then I saw some experiment of ASIM. Yes, so anyway, so value of the BRCP partnership is great for me and help me a lot in my career and ongoing study I will talk about. So the specifically outcome of this grant is summarized in one paper. We published this paper in Cell Report recently. And in this study, we are working on a monkey while moving list flexion extension task. And specifically, we are interested in how, cortical cumulate pathway, I will describe later, is modulated. But we are working with monkey, but Amon, at that time, he already published a paper in Science about mouse version of this pathway. So, mouse has many cutting edge technique is developed and it can be useful in mice, but monkey. So, but he know more about the detailed pathway about this, sensory gating, but we keep discussing to compare species difference between two animals. And my specific hope is to find a mechanism or pathway which is specific for the primate, not in a rodent. So the other stuff I have to include in this talk is how to identify the US collaborator for Japanese researcher. So in my case, as I told you, Amon is working on a rodent and I'm working on the primate. But both of us have a pretty much same scientific interest. That is how brain change controls the sensory gain during the behavior. So one thing I can recommend is you should find the people which have a same, scientific question, but a little bit different between us. That will make us more easy to collaborate without any competition, but more, advantageous each other. So that's my, suggestion. Same question, find a person who have a same question but a different subject. So, but not only science, but to grow the meaningful interaction, it's very important to interact each other, not only scientific field. For example, here I invited Ayman to the Japanese Neuroscience meeting, and here we organize a symposium at the SFN, and the other, we are belonging to a more small community, research community. And at that time, many times we meet together and discuss about many shared interests. So, and sometimes he visit to my student and discussing, and sometimes we go for beer or we are talking about non-scientific issue. So meaningful collaboration, it takes a long time. So you have to have a same scientific question, but also you have to get to know each other slowly. That is a way. So my recommendation is to find a rather small community, not SFN, but more small community, and discuss in many occasions. So that is my second comment. So anyway, let's talk is about science. I'm working on a sensory attenuation during voluntary movements. So this is one example of my research topics. So this is a kind of box which trickle palm of subject hand. Trick is in this case, sometimes subject move this lever, or other experimenter move this lever. So if they measure the trickness or brain activity, when you move yourself, brain activity or tickleness is much smaller than the others one. So somehow your voluntary movement, your own movement is more largely suppress your sensory input outcome by the coming by the voluntary movement itself. So this is so-called sensory gating. So Miller and Daniel Wolpert already proposed that a circuit mechanism, computational mechanism of the sensory gating. Imagine that this is your brain. Your brain sends a motor command to the muscle, and muscle induces activity, movement, and movement itself induces the sensory feedback to somewhere in the brain. But motor command, on the other hand, simultaneously sending a equivalent same command brain signal to and for the model again somewhere in the brain, and that will also send the signal to the neural comparator. So at the neural comparator, brain can subtract motor command you exerted and also sensory consequence of the motor command. So if this is perfectly matched, this is if this is a bit different between these two, you can induce a trickle feeling because this is not predicted by your motor command. So, sorry. The question is, we don't know yet where in the brain this comparator exists and also for the model. So our question is where in the brain your comparator exists. So to ask this question, we train monkey to perform the flexion and extension task. This is an old task. Many people use it. And just a moment, too loud. Okay. So, but this display monkey is watching. Rest period monkey do nothing, and this time Q signaling monkey to move either flexion and extension. And after delay period, monkey have to move as soon as possible to hold, and then 3 seconds to a 3 second hold, monkey release the relax the hand. Then manipulator will automatically return this to the center. So monkey get the reward. So we train this task, and then we explore where is the neural comparator. So this is an anatomical pathway of the sensory ascending system of the somatosensory signal. So primary afferent signal coming cutaneous afferent and muscle spindle, or first into the spinal cord, and then directly or indirectly climb up to the cuneate nucleus, and then second order neuron going to the thalamus and the cerebral cortex. Okay, so sensory gating should be somewhere in this pathway. So first we examine that cerebral cortex. So this is the experiment. We implanted a nerve, so-called nerve cuff electrode into the foreign cutaneous afferent and stimulate there for constant timing, like 3 Hertz during the task performance. So this is EMG, and this is the force wrist torque. So you can see that when you stimulate during the rest period, you can get some nice field potential, but during the movement is suppressed. So this is a so-called sensory gating, and you can easily visualize. You can easily visualize that sensory gating, rest, delay, movement. You can see the field potential is virtually disappear in the cortex. we recently further work on the this phenomena in the subcortical area in M1, area 3B and area 2, and all of this area. This is example, but showing that sensory gating occur during rest. You can see the short latency activity responses gated out here in all area. So in the cellular cortex in S1 and M1, actually more larger area all shows the sensory gating. So that is index that sensory gating may occur cortex or more earlier relay station like a QNet and spinal cord. That is motivate us to record from the QNet nucleus. But QNet nucleus is very, very deep in the middle, actually just a border between brain and spinal cord. So that's the reason why no one ever tried to record there from monkey performing a task. So we tried it. So this is a extracunulate nucleus, which is exclusively, rather expressly relay the proprioceptive input, and this is a main cunulate nucleus is for the tactile input. So how we did it is we use a hybrid image of the CT and MRI. So you take a CT image, and also you took a CT and MRI, and then we hybrided, fused to image. So by doing this, you can plan the experiment how the angle of electrode, how deep that target structure is, and then after establishing this experiment technique, we actually targeted the main cunulate nucleus, but still it's very difficult because cunulate nucleus is a rather complex nucleus with several sub-area. But finally, when we try to main cunulate like green area, we precisely locate electrode, and we could now evoke the sensory evoked potential like this. So by using technology, this technology, we repeat the same experiment like I show you in the cortex. Then we see that during the restful period, SEP field potential is bigger, but during movement, it's rather getting smaller, sensory gating. It's statistically significant. In total, to monkey, you see that this rest field amplitude of the SEP during rest and movement hold. You see that this is a sensory gain modulation, sensory suppression occur also in the cunulate nucleus. But more importantly, if we measure the onset of suppression relative to the movement onset, it's started much before the onset of movement. That suggests that this gating is occurring not from the reafference or peripheral afferent, but descending command also. Descending command sending controlling muscle activity also gating the sensory input. So we further moving on to the single unit activity. This is a trisynaptic short latency response from the induced by the nerve stimulation. And this neuron is active during passive movement. A monkey just resting a hand, and the experimenter just moved manipulum. So you can see some modulation, but more importantly, there's no sensory suppression during rest period, during movement, hold period. No significant changes occur. But active movement, as I show you that, like in LFP, you see that during movement, it's largely suppressed. It's largely suppressed. So that means that both in LFP and single unit, it clearly suggests that sensory gating is occurred in cunulate nucleus. So this is a population analysis, and also as in a population analysis, sensory suppression already started before the movement onset. Yeah, so then we explore the cunulate nucleus and cellular cortex, and cunulate also both show the sensory gating. So the other relay is the spinal cord. So there we also explore the spinal cord. So we implanted a nerve cuff to the spinal cord and then recorded a single unit recording like this. So in the spinal cord, if you measure the cord dozen potential like this, latency of the spike from relative to here suggests that this is a monosynaptic connection from the primary afferent. So we focus on the neuron which have a direct response from the afferent input. So then this is the result. Again, during this is the amplitude of the response. During the active movement and hold period, is sensory gating occur. So in short, spinal cord is also showing the sensory gating. So then so far, we found that the sensory gating occur in the spinal cord, cuneate, and cortex. But we may compare the size of gating. It's we want some interesting trend. So in spinal cord, it constantly suppressed by 20% of the original amplitude. But cuneate nucleus, it decreased much larger, 60%. And cortex, also 60%. What that mean? So maybe even if people measure sensory gating in the cortical LFP, that is already occurred in cuneate nucleus or spinal cord. And cortical field potential just represent these sensory gating, not actively modulating the incoming sensory signal within the cortex. So then our conclusion is a neural comparator is exists in the cuneate nucleus and the spinal cord. And then I would like to show the preliminary data, which is actually main target of our third grant of this grant framework. So in this case, as I told you, Ayman already published a paper about the corticocuneate gating. So we would like to compare this system and the primary system. So first, we inject the anterograde tracer into the cuneate nucleus and see where in the brain actually project to the cuneate. So then we successfully injected the main cuneate and extra cuneate, and then mapping out the cortical leveling. So all of these neurons have a direct projection to cuneate nucleus, as Ayman showed in the rodent. So then this is the result. So most interesting part is, okay, so this is a motor cortex. This is sensory cortex, primary sensory cortex and higher sensory cortex. So what interests for us is a new M1. Motor cortex, all of animals have a, relatively have a motor cortex, but new M1 is within the central sulcus. It's highly developed in the only primate. Okay. Okay, only primates. So this is a main difference between the rodent and primate and rodent and primates. And anyway, so also there's a projection from the higher sensory area. So this is also not existing in the primate rodents. So this is another trend. So anyway, main projection is coming from the sensory primary and higher sensory area. So that will be the source of the sensory gating. So then we are now injecting anterograde tracer into there and leveling into the area 2, area 3, and area 4, and finding the terminal level in the cuneate nucleus and the spinal cord. And this is the target. Then we are now doing the in situ hybridization of the spinal cord to see if this area is receiving cortical command. If this area is responsible for the sensory gating, we should find a cluster of inhibitory neurons. So we are now working on that. And then finally, after we identify the pathway, we will do the direct experiment to selectively suppress this pathway. So selectively eliminate sensory gating. And what is happening after that? That is our ongoing project. So conclusion is tentatively conclusion is for the model is a new M1 and S1, and also this is a source of the this can be a main source of sensory gating in the primate brain. So this is my laboratory, and especially this person is contributed a lot for this work. Thank you very much.
Thank you very much. Very fascinating talk. And so do we have time to accept one question, quick question, or later? After. Okay. Thank you very much. And hopefully Dr. Seki will stay with us and we have some time to ask him questions at the end. Thank you very much. And I switch to Dr. Benson.
Yes, thank you, Dr. Seki. Excellent presentation. Yeah, I have a couple of questions for afterwards. But I'm going to introduce our next speaker from the US side. This is Dr. Weidong Chi. Dr. Weidong Chi is a clinical associate professor in the Department of Psychiatry and Behavioral Sciences at Stanford University School of Medicine. Dr. Chi earned his PhD in cognitive neuroscience and conducted functional neuroimaging research of cognitive control in Dr. Hong Chong Luong's lab at the SUNY Stony Brook. He further gained experience during his postdoctoral training in Dr. Adam Aaron's lab at the University of California, San Diego, where he investigated the dual mechanisms of action control using transcranial magnetic stimulation and functional MRI. Currently, Dr. Chi's research is centered on uncovering the brain mechanisms underlying cognitive deficits in two key populations: children with neurodevelopmental disorders, especially those with ADHD, and elderly individuals suffering from neurodegenerative diseases such as Parkinson's disease. By integrative cognitive neuroscience and computational models with advanced functional neuroimaging techniques, Dr. Chi's research aims at elucidating the neurocognitive effects that drive atypical brain development and pathological aging. The title of his talk, Dr. Chi is funded through the National Institute of Mental Health. This program, this US-Japan bilateral program, we have 5 other of our NIH partners that work with us. And so he's funded through the National Institute of Mental Health. The title is Methylphenidate Modulates Brain Dynamics in Children with ADHD. Dr. Chi.
Thank you, Richard, for a kind introduction, and thanks for the invitation. And it's my great pleasure to be here and talk about this collaborative research. So I have no disclosure. So ADHD, attention deficit hyperactivity disorder, is one of the most common neurodevelopmental disorders in the world. So in the United States, there has been an alarming trend with increased cases. So a study published about 5 years ago found that ADHD prevalence rate in children and adolescents has nearly doubled in the last 2 decades. So as a result, a national survey conducted 2 years ago revealed that a prevalence rate based on the parent report, ADHD is over 10%. And if we take a close look at distribution of data by age and gender, you can see we know boys has more likely to be labeled as ADHD than girls at a younger age. So if you look at the age of 10 years old, there's nearly 20% of boys may have ADHD or have behaviors. So it's quite striking, right? So in Japan, so the prevalence rate is not as high as in the United States, but still over 6%. And a new study found there's an increased trend of new cases in children in Japan as well. So the disorder has become a public health issue in the United States, Japan, and other countries in the world. So it's a global impact. So ADHD is characterized by behavior problems and clinical symptoms such as inattention, hyperactivity, and impulsivity. And behavior studies have revealed that children with ADHD shows deficit in performance like in cognitive control task. So cognitive control is a very broad term. It refers to a high order function for us to coordinate our different cognitive, affective, and motor process and to deal with conflict in order to achieve some short-term and long-term goals. And a study have revealed like kids with ADHD show like a lower accuracy, slow response time, unstable behavior. So including our own study, which we show that children with ADHD, they are much slower in controlling their impulsive responses. And also sometimes after they're making mistakes, they do not adjust their behavior as much as the typical developing kids. So it suggests that they may have some deficits in detecting the salient signals such as errors, and also have difficulty in regulating behaviors after making mistakes. So what is the brain systems underlying cognitive control? So decades of neuroimaging research and also meta-analysis show that there's a set of brain regions that consistently activate in different kind of control tasks. Those mostly including the salience and the frontal parietal networks. So salience networks mostly including the anterior insular and anterior cingulate cortex, which are a system important for detecting behaviorally salient signal such as error. And also the frontal parietal networks, mostly including the dorsolateral prefrontal cortex and posterior parietal cortex, which is a system well known for its role in working memory, holding and updating important information for upcoming decision making. There's a third default mode network, which consists of posterior cingulate cortex and ventral medial prefrontal cortex. So this system shows deactivation rather than activation during cognitive mind task. However, its interplay with the salience and the frontal parietal network has been shown to be important for cognitive control. So we are focusing on these three networks in the cognitive control systems. So actually, the neuroimaging study from clinical research, I already showed like children with ADHD, they have shown like abnormal activation in salience, frontal parietal, and default mode networks when they are engaged in cognitive control tasks. So it suggests that maybe the dysfunction or dysregulation in these three systems may undermine the cognitive function in affected children. So medication has been widely used to treat ADHD symptoms. It cannot cure disorder, but it can help reduce the symptoms. And the study has shown like in addition to reduce the clinical symptoms, it also has positive effect to daily function. For example, can improve school performance, can reduce accident, self-injury, suicide behavior, and also has beneficial effect in mood disorders. So methylphenidate is one of like the mostly common used stimulant for treating ADHD. However, we don't know that much about how the methylphenidate working on the brain and has a downstream effect on behavior. So this research, we're trying to understand how methylphenidate medication impact brain dynamics in ADHD kids. We focus on dynamics because RENA study has shown like human cognition and cognitive dysfunction really relies on a stable and a flexible brain dynamics. So we try to focus on this front, which is less studied in our research. So we have three broad goals. One is to look into the medication effects on behavior, cognitive control. The second, look into medication effect on the brain dynamics. We specifically focus on cognitive control and the reward systems. The last is look into the brain behavior association affected by the medication. So the study is conducted by my collaborator, Dr. Yoshifumi Mizuno, and his wonderful team in Fukui University in Japan. They recruit about like 30 children with ADHD and about 60 TD controls. So every kid will come to do the resting fMRI study and also auto scanner and behavior task. For each kid with ADHD, they have 2 visits. In one visit, they come and take methylphenidate pills. In another visit, they come to visit and have taken a placebo pills. So it's a control for medication. And the order of the methylphenidate and the placebo are counterbalanced across subject. So the order is not confounding back here. So in every visit, they will do a resting fMRI scan for 6 minutes. Out of a scanner, they're doing a go-no-go task. Basically, there's a group of stimulus, a go stimulus, a no-go stimulus. For go stimulus, the subject needs to press a button to make a quick responses. For no-go stimulus, they have to control their tendency to respond. They have to inhibit. And behavior such as error rate and RT and variability of response will be used to measure as attention. So first, we replicate previous finding showing that methylphenidate has positive effect in behavior. And this figure shows you the error rate and RT and RT variability. Basically, the higher score is a worse performance. As you can see, the methylphenidate compared to placebo, it reduces the error rate and make response faster and more stable. So next, we want to get understanding about, in general, how the medication affects both signal in terms of fluctuation at the control system and the reward systems. So we use a measure called amplitude of low frequency fluctuation, which basically measures the variability of both signal in fMRI time series. And what we found is the medication has enhanced the fluctuation in the Sealan's network and the default network, which is a core system for the current control. We also found the medication enhanced the spontaneous activity in the right nuclear compounds, which is a keynote in the reward circuitry. Next, we want to look into more detail in brain dynamics. We want to see from moment to moment, is there a certain brain dynamics and how dynamics are impacted by the application. So we use a method called Bayesian switching dynamic system, in short BSDS, which is a method developed by my former colleague, Dr. Lita Hill. This is basically an unsupervised learning method, try to identify latent brain state based on the unique pattern of activity and connectivity between brain regions from fMRI time series. So in our previous study, we have validated a method using the simulation data and the rodent optogenetic fMRI data, which we know the ground truth of this data. So we construct 11 regions of interest, including the ROIs in the Sealan's and frontal parietal and default network. We extract time series from its ROIs and apply the algorithm. So the BSDS uncovered 5 different brain states. So let me help you understand these figures. So in each row represent one subject, and each column represents a time point or the fMRI scan volumes. So, and a different color represent a different state defined by different pattern of connectivity and activity. So here, there's 5 different states, S1 to S5. So this figure basically shows the temporal evolution of different brain state from time point 0 to time point n for every subject. So then we ask, we can look into the temporal profile of the brain state within subjects and compare between subjects. We ask whether medication have impact in dynamics, and also dynamics is different between ADHD versus controls. So here I show you bar figures is by like a number of state and occupancy rate, how often the state occur, and a different color represent different group. So the blue is TD controls, red is ADHD in placebo condition, and brown is the ADHD in methylphenidate conditions. What you can tell is in the specifically state S2, there's an increased occupancy rate in the placebo condition compared to TD, suggesting children with ADHD has much higher occupancy rate. So this state happens a lot for kids with ADHD than controls. Interestingly, methylphenidate reduces this hyper occupancy rate in kids with ADHD. Actually, it brings down to a level very similar to controls. Then we can look into the spatial property of the dynamic brain state. So specifically for this state S2, we first compare placebo condition versus the controls. We see there is increased connectivity between posterior cingulate cortex and the ventral medial peripheral cortex, which are 2 key nodes in the default mode network. So this suggests ADHD kids have a hyper connectivity within the default mode network compared to controls. And methylphenidate can reduce the connectivity within the default mode network, but at the same time, it also reduces connectivity between default mode networks and other regions in the frontal parietal systems. So last we asked whether the effect medication on the brain connectivity can associate with the effect on the behavior. So here we use the behavior measure as the intra-individual response variability, basically measure the fluctuation of response time as a measure of attention. So what we see here is specifically the medication effect on the connectivity between left anterior insular and the posterior cingulate cortex, which are two keynotes for the Silience network and default mode network. So basically, the medication effect on the connectivity between Silience and default mode networks is associated with medication effect on attention. And this is also consistent with our previous study in task fMRI data. So in summary, our work suggests the melatonin has positive roles to normalize the aberrant dynamics in children with ADHD, and the medication effect in connectivity can predict the behavior changes induced by medication. So all this work are impossible without my wonderful collaborator and his teams in Japan in Fukuoka University. And here is a picture of his team member, and here's the location of Fukuoka University in Japan. So Dr. Mizuno has a lab at Stanford for 2 years. So we collaborate on analyzing data and to get a more interesting idea from a data set. So in addition to the study, I'm just telling you, so Dr. Mizuno also lead a project called Children Developmental MI Project. This is a large-scale data collection, including the MI data, behavior data, eye tracking, and genetic neurotransmitter data. So we hope to continue our collaboration to this front to get more understanding in the typical and atypical development. We also hope we can integrate the Japan data set with the data set in the United States, for example, the ABCD study, as well as the data set from other countries to enhance the robustness test and reproducibility, generalizability of our findings. So last, I want to thank my colleague and the collaborators at Stanford and other university, and as well as the funding resource, especially at NIH, and also the many large-scale public data set. It's so important for research now, and thanks for your attention.
I think if there are any questions, we actually have a microphone here in the center, or you could just raise your hand. It's sort of a smaller, intimate crowd for either speaker. Please either raise your hand or come up to the mic if you have a question, or I guess anyone online as well if you have a question. I did have a question for Dr. Siki. Both excellent presentations that we had today. My question is about, when you think you have certain reflexes that occur, sort of if someone receives noxious stimulation, that they can sort of withdraw without that cortical input sort of modulating that. With this stimulation of the hand in the monkeys and making the movement, does that attenuate over time? And is there, does that, through that constant stimulation, is there a cortical mechanism to stimulate the response over time? And then is there sort of a cortical component as well that sort of that sort of modulates this, that's in a separate area from the primary motor cortex region? If you want to come up here.
Yeah, I think your question is about when you stimulate here, brain specifically suppress at timing or more constantly suppress. That is depend on the situation. You know, if monkeys habituated this stimulation, and this information from palm is not so crucial for the behavior, monkey try to suppress continuously. But something is also important for monkey to perform task from here, then it's more timing suppression occur only that specific timing. Monkey predict this timing, I will be perturbed. But if simulation is random, then monkey cannot predict. In that case, monkey suppress every time.
So if it's random, then they will suppress it. Okay, no, thank you. And I did have a question for Dr. Chi as well. It's interesting. You mentioned that ADHD, it's increasing in the US, well, globally, and Japan. And any reasons for that? Is there something that's leading to this this problem? Why do you think it's increasing?
Yeah, thanks. That's a great question. So we don't really know all the causes for this increase, and there's multiple guesses. One is like education. So compared to like 20 years ago, people know more about ADHD, the disorders. The parents know about that, and also like maybe there's environmental factors. So, but there's many things we don't know. Yeah.
Thank you. So I believe it's been recently raised up. It's been talked about in the HD context, some difference between boys and girls, and how I read a little bit about how in girls it can be you can see a little bit of a case of misdiagnosis. And so I was wondering if you noted in your experiments administering an MPH, if you noted any difference between the behavior between boys and girls and how the medicine is affecting them. Thank you.
Yes, great question. So I don't know much about like how the medication affected the girls and boys differently, but that's certainly a big sex differences. So one is like at a younger age, boys are more likely be labeled as ADHD compared to girls. That's partly because like boys more like to intend to be hyperactive, and the girls more like inattention type. So it's more easily be picked up by teachers in the classroom. But more and more evidence suggests that there's like underdiagnosis of ADHD for girls, and at older age, like close to adults, more and more females will think themselves maybe have ADHD. So there's a trend of increase of females of disorders when getting older. And there's a lot of research in this front to look into sex differences in terms of behavior, medication effect, and the medication effect. I was talking about like a medication effect is effective to reduce the symptoms, but there's a lot of variabilities in terms of whether the person will be responsive to certain medication and to what kind of medication. So there's lots of differences there. Yeah.
Any other questions? Okay. Well, the purpose of this, as I mentioned, that we have this program, NINDS, the Neuroscience Institute at the NIH. We lead this, but we have 5 of our other sister institutes that also work with us that help to support this program. We'd like to increase, and I know this was brought up, we'd like to increase the collaborations between the US and Japan. And so we're definitely open to suggestions of how we can get the word out about this program to get more collaborations developed. This will be sort of one of the topics as well during our strategic planning process as we think through how to do this more, partnering on the NIH side, as well as working with you as well, our partners in Japan. I don't know if you have any final words, Dr. Sada, you'd like to make?
So I'd like to thank the 2 speakers for the wonderful talks. And also I'd like to thank everyone here in the audience as well as online for joining us today. I hope that the 2 talks will stimulate and encourage new collaborations and partnerships between neuroscientists from the United States and Japan. Thank you very much.